We often learn that diffusion is a simple and predictable process. Imagine a drop of ink spreading in water or sugar dissolving in tea. Over time, the particles just spread out evenly. This classical idea says that the longer you wait, the farther particles travel, and how far they move should grow steadily with time. But here's the paradox. In many real systems, especially in biology, soft matter, or even space, uh, this simple rule does not always apply. Sometimes particles spread more slowly than expected. Other times they move faster than what traditional diffusion would predict. This unusual behavior is called anomalous diffusion, and it raises a deep question. How can the same basic physical process, random motion, give rise to such different outcomes? Let's break it down. In slower than normal diffusion, also known as sub-diffusion, particles get stuck, delayed, or held back as they try to move. This happens in crowded or complex environments, like inside a living cell. Think of a person trying to walk through a tightly packed crowd at a concert. You cannot move freely, you bump into others, backtrack, and wait for space to clear. Molecules inside cells behave the same way. They encounter obstacles, get trapped in molecular cages, or are temporarily bound to structures. Their progress is no longer smooth or predictable. It becomes sluggish and history dependent. Now consider the opposite. In faster than nominal diffusion or super-diffusion, particles don't just move randomly. They make occasional, very long jumps or move with bursts of energy. Imagine a person running across a park, but now and then teleporting ahead 10 meters. Some particles behave like that too, especially in turbulent fluids, active materials, or environments with strong energy bursts. This happens in systems like bacterial swarms, plasma in space, or even animal foraging patterns, where creatures take a mix of short steps and sudden leaps. Why does this happen? Shouldn't randomness always even out? Here's the key idea. Not all randomness follows the same rules. Traditional diffusion assumes that each step a particle takes is independent of the last, and that the time between steps is fairly uniform. But in the real world, steps can be correlated, meaning one step influences the next, or the waiting times can vary wildly. In some systems, a particle might get stuck for a long time before suddenly moving again. In others, the particle keeps moving in the same direction for a while, building momentum. These subtle changes in behavior, repeated over many steps, completely change how the particle spreads over time. So where do we actually see this in nature? In biology, scientists have used high-resolution microscopes to watch individual molecules move inside living cells. These molecules often slow down and speed up in irregular ways, clearly defying classical predictions. In ecology, animals like sharks and birds use a mix of short, frequent moves and rare, long-distance flights, a strategy that mimics super-diffusion and helps them search more efficiently. Even in our brains, some signals and molecules travel in ways that hint at anomalous patterns. But if diffusion does not follow the standard rule, how do scientists study and describe it? Rather than relying on fixed speed of averages, researchers now use flexible models that take into account memory effects, pauses, traps, and occasional jumps. These models allow scientists to better understand how things move in complex systems. Whether they are drugs navigating tissue, nutrients traveling in plants, or chemicals diffusing in a gel, why does all this matter? Because if we misjudge how something spreads, we can make critical errors. Imagine designing a medicine based on the assumption that it will reach a tumor quickly, only to find it gets trapped along the way. Or imagine tracking pollution through soil, only to find that it moves faster or slower than expected. Anomalous diffusion helps us refine our expectations and design smarter solutions, especially in fields like drug delivery, material design, environmental science, and even finance. In the end, the diffusion paradox teaches us that simple ideas often need to be revisited when we explore complex realities. Particles are not always polite little wanderers. Sometimes they hesitate, sometimes they leap. And when they do, they reveal new rules of motion hiding in plain sight. The diffusion paradox describes how, in many systems, particles do not spread out as predicted by classical diffusion theories. Instead of moving steadily and smoothly over time, some particles move either much slower or much faster than expected. This unexpected behavior occurs in both living cells and non-living systems like porous rocks or turbulent air. It challenges the idea that all random movement follows the same universal rules. The paradox highlights that nature is often more complex than our simplest models. Scientists observe that proteins in the interior of living cells move much slower than they do in a test tube, defying basic expectations. It's like watching someone on an escalator who sometimes gets stuck and other times suddenly shoots ahead. Very different from steady walking. In classical diffusion, particles are expected to wander randomly and cover more ground steadily as time passes. Each step is assumed to be random, and the environment is assumed to be the same everywhere. The theory says that the more time you wait, the further a particle should travel on average. These rules come from simple observations of things like dye spreading in water or gas filling a room, but they only work well when the surroundings are uniform and there are no obstacles or memory effects. When you place a drop of food coloring in a glass of water, it gradually spreads out in a smooth, predictable way. It's like predicting how far a hiker will go if they take steps of equal size in random directions across a flat field. In crowded or sticky environments, particles often get delayed or trapped along the way. They may pause in one place before moving again or encounter tiny barriers that slow them down. This causes them to move less than expected over a given period. These slow-moving behaviors are commonly seen in gels, thick liquids, and the interiors of biological cells. The result is a spreading process that's slower than classical models would predict. Inside living cells, molecules like enzymes often get caught in dense networks of proteins and other molecules, slowing their movement. It's like trying to walk through a packed crowd. You keep stopping and starting, so you travel much less than you would in an empty hallway. In other cases, particles move in a way that seems too fast for normal random motion. This can happen when particles are pushed along by internal energy sources, like tiny motors or chemical reactions. It can also happen when particles make occasional very long jumps, covering more ground than usual. These leaps or persistent directions allow particles to travel faster than expected over time. Such behaviors are common in active biological systems and chaotic flow. Certain bacteria use rotating tails to propel themselves through fluids, allowing them to move farther and faster than random drift would allow. It's like someone riding a skateboard down a slope instead of walking. They go much farther in the same time. Real environments are not always smooth and even. Many have holes, blockages, or different types of materials in them. When particles travel through such varied spaces, their movement becomes irregular. In some regions, they may pass quickly, while in others, they may get delayed or stuck. This unevenness changes how far particles travel overall, making the process faster or slower than expected. These environmental effects are a major reason diffusion behaves differently in real systems. Oil trapped in underground rocks often moves unpredictably through a maze of channels and dead ends. It's like trying to drive through a city with both highways and narrow alleyways. You speed up in some places but crawl in others. In classical diffusion, every step a particle takes is completely independent of the last one. It has no memory. But in many systems, the way a particle moved before can affect how it moves now. This can happen when particles are dragged by elastic forces or slowed down by past interactions. Such systems behave like they remember their own history, and this changes how the motion unfolds. These memory effects make the whole process more complicated and less predictable. Tiny beads inside a gel tend to slowly return to their previous positions after being pushed, as if pulled by invisible rubber bands. It's like stretching a spring. Once you let go, it snaps back, showing that it remembers how it was stretched. To describe these unusual types of movement, scientists have developed new types of models that go beyond the classical rules. Some models allow particles to wait for random periods before moving again. Others describe the chance of taking very large steps that happen only once in a while. Some models even account for the way particles are influenced by past steps or energy input. These new tools help researchers explain the strange diffusion seen in many systems. Physicists use a model called a random walk with traps to explain how particles pause for long times in sticky environments. It's like updating the rules of a board game so that a player can either rest for several turns or suddenly jump ahead many spaces. Understanding these strange patterns of motion helps scientists improve technologies in medicine, materials, and environmental science. In biology, it explains how drugs spread through tissues or how cells communicate. In industry, it's used to optimize how fluids move through filters or how pollutants move underground. If engineers don't account for these effects, their designs might fail or perform poorly. That's why the diffusion paradox is more than just a theoretical curiosity. Drug molecules often move unpredictably through tumors, which affects how effective cancer treatments can be. It's like trying to deliver a package on time, but the streets in the neighborhood are full of unexpected roadblocks and shortcuts. Scientists still don't fully understand why some materials or systems show such extreme differences in particle motion. Ongoing experiments try to map out which kinds of environments cause slowdowns or speedups. New tools like super resolution microscopes and single particle tracking are helping to uncover hidden behaviors. Researchers are also exploring how these ideas apply to the quantum world or even economics. Solving the diffusion paradox could lead to better models for everything from brain activity to planetary atmospheres. Experiments with nanoparticles show that their motion changes dramatically depending on whether they are inside a cell, in water, or near a surface. It's like watching a fish behave completely differently in a pond, an aquarium, or the ocean. Same fish, different world.